second year fellows. So just some basic overview. We'll start by talking about what is cerebral palsy. It is a major non-progressive neurodevelopmental disorder affecting early motor development. Characteristically, it impairs movement and posture. It is frequently seen with intellectual disability, epilepsy, and autism spectrum, and it affects 2.6 to 2.9 per 1,000 deliveries. In terms of the etiology, I feel like when I was an intern, everyone was like, all of the cerebral palsy is caused by poor management during labor. And that's actually not really true. It's the result of injury to the developing brain that occurs during the prenatal, perinatal, or postnatal life. And 75 to 80% are attributed to events during the pregnancy before labor. 10% of them are due to interpartum events such as birth asphyxia, and another 10% was due to postnatal things like head injury or CNS infection. And current estimates indicate that about 30% of CP cases may actually be genetic in nature. So in terms of risk factors, there are many. Prematurity, multiples, growth restriction, intracranial hemorrhage, periventricular leukomalacia, which adversely affects the white matter of the brain and develops likely between like 24 to 36 weeks. This causes damage to the white matter, the cells then die, and this creates empty space in the brain, leading to like spasticity, cognitive impairment, and vision issues. There's also the risk of infection, placental pathology, structural brain abnormalities, and then finally genetic syndromes, which is going to be the focus of this talk. A lot of the times the origins tend to be multifactorial, but sometimes no cause is clearly identified. So registry-based data in terms of genetics shows that about 21 to 40% have an associated congenital anomaly. And there's an increased risk in consanguineous families and monozygotic twins, which makes sense. And 40% heritability estimated in probabilistic modeling in a Western Swedish cohort study. And then the associated congenital anomalies implicate like genomic alterations in many cases. In terms of the etiology, this is, I thought was just like a nice figure to kind of think about something that I don't really think about every single day anymore in terms of genetics. So we have our mitochondrial DNA, which we know is primarily from mom. Then we have our nuclear chromosomal DNA, which is from both parents. This can lead to issues with copy number variants, as well as epigenetics, which is like the methylation that we sometimes think about. And then there can also be an issue with individual gene mutations in the nucleotides themselves. And any of these four places can lead to loss of normal cellular function of the proteins that are encoded by these genes. This figure just kind of goes over effect size. So the effect of a given genetic mutation may vary depending on the nature of the mutation and the presence or absence of environmental insults and the individual genomic context in which a mutation occurs. A severely deleterious mutation may lead to a major effect size, while a less damaging mutation that does not disrupt the protein function as profoundly may lead to a smaller effect size. So accordingly, a highly damaging mutation can be sufficient in and of itself to cause cerebral palsy in some individuals, whereas in other cases, the additive effect of the less damaging mutation coupled with an environmental insult such as ischemia may cross the threshold needed to produce neuromotor disability. And then also in some instances, a single gene alone will not lead to clinical CP, but the cumulative effect of several less deleterious mutations acting together in a polygenic fashion can lead to the disorder. So in terms of kind of like broad strokes of how we think this happens, we have induction of the hypoxia signaling pathway that leads to activation of hypoxia-inducible transcription factors. So that's like 1 and 2. And these factors have been shown to trigger expression of genes involved in oxygen transport, glycolytic metabolism, angiogenesis, cell cycle control, cell proliferation, apoptosis, and energy metabolism. There's also the involvement of microRNAs, which are non-protein coding RNAs made up of 20 to 24 nucleotides that mediate hypoxic stress response because they are able to modify gene expression both rapidly and reversibly. And this ultimately leads to chronic fetal hypoxemia, leading to a fetal inflammatory response, causing the release of a cytokine cascade and activation of apoptosis, leading to cell death. Another way of looking at this is in this figure. So you have your gene susceptibility leading to, and then you can also add in your number, duration, severity, and timing of insults. You can have other things like growth restriction or issues with placental function. And ultimately, this leads to the affected neuronal, structural, and functional integrity being affected. And hypoxia is often a compounding problem in a fetus that's already affected by a genetic condition. There was one study that of just babies with Prader-Willi that 23% of them were also complicated by birth asphyxia. Similarly, the rate of intrapartum complications, including sustained cyanosis, the need for oxygen or ventilatory support, cardiopulmonary resuscitation, prolonged NICU admission, is significantly higher in children with malformations of cortical development compared with a control group of children that also just had idiopathic epilepsy. The profound multigenic control of the brain structure and function most likely explains the range of reactions encountered after hypoxia. And fundamental genomics also may serve to explain how the frequency of cerebral palsy hasn't really changed in the Western world, despite our using electronic fetal monitoring and sophisticated technology and a pretty high C-section rate. So in terms of the genetics, I kind of broke it into these 4 categories. So we have our single nucleotide polymorphisms, susceptibility genes, copy number variants, and monogenetic causes. So first, I will talk about the single nucleotide polymorphism. So a single nucleotide polymorphism is a variation at a single position in a DNA sequence among individuals. If more than 1% of a population does not carry the same nucleotide at that specific position in the DNA sequence, it's considered a single nucleotide polymorphism, or a SNP, SNP. If a SNP occurs within a gene, then the gene is described as having more than one allele. And in these cases, the SNP may lead to variations in the amino acid sequence. SNPs, however, are not just associated with genes, they can also occur in non-coding regions of DNA. Initially, this was used to identify genetic variations that were found to be in significantly higher frequency in cerebral palsy cohorts compared with controls. So SNPs involved in inflammation and coagulation have been implicated as a risk factor for cerebral palsy. More than 20 studies of CP have evaluated alterations in genes that regulate the inflammatory and coagulation cascade, and polymorphisms in more than 15 genes have been associated with CP. However, as with most genetic association studies, they're hampered by multiple comparisons, small sample size, population heterogeneity, leading to inconsistent findings. So I'm just going to go through a couple different examples. One that kept coming up in the literature was IL-6, and this is the promoter region polymorphism. And IL-6 is associated with an increased risk of cerebral palsy in 2 separate populations. This suggests an altered fetal inflammatory response that is due to genetic variation that is contributing to an increased risk of CP. Another one that keeps coming up in the literature is alleolipoprotein E. This is a little bit more of a confusing one. It was initially thought to be protective against cerebral palsy, but ApoE has actually been found to be associated with a worse phenotype in one study, but this was later not confirmed in others. And additional studies found that there was increased risk with the E2 and E3 allele. It's found on the Q arm of chromosome 19. It's essential for catabolism of triglyceride-rich lipoprotein constituents. And we see that there's increased expression in the liver and kidneys, and it's associated with familial hyperlipoproteinemia type 3 and ischemic strokes in adults. And the thought is that it plays a role in cardiovascular disease and it may predispose to CP by perinatal stroke. Next, we have nitric oxide synthase. It was initially evaluated because of its role in ischemic and inflammatory brain injury. And overexpression has been reported in newborn periventricular white matter injury and adult stroke. It's a key mediator of oligodendrocyte injury in the newborn brain injury due to intrauterine infection. And the polymorphism associated with CP was seen in 2 separate populations. Next, we're going to talk about osteopontin. It's a soluble immune factor shown to promote axonal regrowth and synaptogenesis following injury. Pro-inflammatory is a pro-inflammatory cytokine secreted from activated macrophages and T lymphocytes. And it's due to the single nucleotide polymorphism, and that this is associated with cerebral palsy. So next I'm going to talk about susceptibility genes. And similarly, this was initially used to identify genetic variations that were found with significantly higher frequency in CP cohorts compared with controls. And the big one I wanted to touch on is the idea of thrombophilia. So factor V Leiden, prothrombin, protein C, protein S, antithrombin. The thought being that these things lead to a prothrombotic state, and there's an increased risk of perinatal stroke, which can increase the risk of cerebral palsy. The influence, however, of these procoagulant factors yielded contradictory results. And a recent systematic review indicates that the link is modest at best, and it needs further evaluation. Next, we're going to talk about copy number variants. So this refers to the genetic trait involving the number of copies of a particular gene presenting in the genome of an individual. Genetic variants including insertions, deletions, duplications of segments of DNA are also collectively referred to as copy number variants. So they're significantly challenging to interpret. The rarity of individual copy number variants leads to infrequent overlap of genomic regions that those variants span. And this can be de novo or inherited from unaffected parents. And it encompasses multiple genes, making it difficult to draw firm conclusions. It's kind of fraught with variable expressivity, incomplete penetrance, higher genetic robustness of female versus male. Presence of epigenetic modifiers or second hits could explain why a parent could be unaffected and doesn't manifest symptoms, but their child may. So we're just going to go through a couple of them. The first one is carnitine delta 2. It's on the 5p arm, or the p arm of chromosome 5. And the copy number variant we see is that there is a 446 kilobase duplication in the first exon. And this gene normally encodes for adhesive junction associated proteins, and it's implicated in brain and eye development. The next one we're going to talk about is STAM1. This stands for disheveled associated activator of morphogenesis 1. It's on the Q arm of chromosome 14. It is a 265 kilobase duplication, and this normally encodes for formin homology proteins, and they're implicated in cell polarity, and there's usually expression in the skin. Next, we're going to move to monogenic causes. So over the last decade, several single gene causes of cerebral palsy have been identified. And this has been primarily by using next-gen sequencing. Somewhat surprisingly, these monogenic causes of cerebral palsy have not mapped to pathways controlling inflammation or thrombosis, which is kind of where we were with the other couple categories. And if one views these genetic mutations as a glimpse into crucial neuronal pathways relevant to CP, an important role for the neurodevelopmental process has instead been highlighted. So first, we're going to talk about the ataxic CP genes. So the big one was KCNC3, and this is a potassium voltage gated channel subfamily C member 3. It's autosomal dominant in its inheritance, and it causes spinocerebral ataxia type 13. It's late onset and progressive condition, and it encodes for the potassium channels, and the mutation that we see is that there's a loss of function mutation. Next, we have KINK1, which stands for KN motif and acrolein repeat domains 1. It is a heterozygous deletion that was first described in a single Israeli family, and there is a cytoskeleton interacting proteins that limit actin filament growth to prevent uncontrolled actin polymerization. It also interacts with microtubule plus ends. We see early neuromotor delay, hypotonia that progresses to spastic quadriplegia, and the intellectual disability can be severe. The brain MRI shows diminished cortical volumes without focal pathology. And the imprinting pattern is complex and kind of incompletely understood, and most recently suggests random monoallelic expression. There's also biallelic mutations that may cause steroid-resistant nephrotic syndrome. Moving forward, additional studies, including functional validation assays of novel variants that may or may not ultimately be confirmed as being important to disease-causing mutations, will be important to clarify the role of KINK1 in spastic cerebral palsy. Next, I was going to touch on adaptor protein 4 complex subunit. So these are mutations in AP4M1, AP4B, AP4S1, and AP4E1. These genes encode for subunits of a heteromeric adapter protein complex, AP4, that participates in intracellular trafficking. This receptor excitotoxicity is believed to contribute to hypoxic ischemic white matter injury and may play a role in subsequent CP. And this AP4 facilitates that receptor trafficking. It's also believed that AP4 functions at the trans-Golgi network. If you guys remember this from biology, bringing in these AMPA receptors to the cell surface via cargo endosomes. And consistent with this model, defective AP4 function would be expected to lead to diminished cell surface receptor abundance. However, further work is needed to determine the role of AP4 in this receptor trafficking, and if this is the main mechanism at work in this form of genetic cerebral palsy. For babies that are affected by this, you characteristically will see dysmorphic features, including hypotonia, intellectual disability, growth retardation, and microcephaly. And MRI will show colpocephaly, which is a congenital brain abnormality in which the occipital horns, which is like the posterior and rear portions of the lateral ventricles of the brain, are larger than normal because the white matter in the posterior cerebrum has failed to develop or thicken. You can also see decreased white matter, cortical and or cerebellar volume loss. And postmortem studies in patients with this mutation have shown simplified morphologically abnormal Purkinje cells and their respective dendritic locations. So next we have ADD3, which stands for Ductin3. This was initially seen in a consanguineous Jordanian family, and it's a homozygous mutation causing spastic cerebral palsy. It interferes with the protein's ability to cap actin filaments, leading to abnormal actin filament accumulation. You can see mild microcephaly and hypotonia progressing to spastic dysplasia. There can be borderline intelligence or intellectual disability, and neuroimaging demonstrates diminished white matter volume. And then next I wanted to touch on a couple patients or a couple papers that discuss future directions. So there are some candidate cerebral palsy genes. Recent studies using whole exome sequencing have identified multiple candidate genes. And this work also suggests that de novo mutations may be significantly contributing to cerebral palsy. These can be heterozygous, so some of them are inherited, some of them are de novo, and there are also X-linked variants that have been found. And lastly, I wanted to touch on this paper that came out in JAMA of this year that did molecular diagnostic yield of whole exome sequencing in patients with cerebral palsy. So the objective was to determine the molecular diagnostic yield of exome sequencing in individuals with CP. It was a retrospective cohort study, and the exposure was exome sequencing with copy number variant detection. And the primary outcome was molecular diagnostic yield of the exome sequencing. So in terms of their study design, there are two cohorts. One was a pediatric patient cohort with cerebral palsy that was referred for exome sequencing from a clinical lab. It included over 1,000 trios, so that refers to a patient with cerebral palsy plus both of their parents. And then the other cohort was 181 adult patients with cerebral palsy that had been recruited from another study. And then the exome sequencing was also performed on them. In terms of what they found, in terms of the molecular diagnostic yield, they found that about 440 of the 1,345 pediatric patients, so around 32%, had a molecular diagnostic yield. And of that 32%, 94.3% of it was due to a single nucleotide variant, 4.3% was due to a copy number variant, and 1.4% included both. So when they looked at the trios and testing of the proband was done concurrently with parents, that significantly improved the overall molecular diagnostic yield from 23.3 to 35.4%. And then if you look at the inheritance pattern, about 72% of it was de novo, 19% of it was autosomal recessive, 5% was autosomal dominant, and 3.4% was X-linked. So the exome sequencing derived copy number variant analysis and identified 25 patients with pathogenic and likely pathogenic CNVs. And these copy number variants range from 637 base pairs to 93.3 mega base pairs. And there were 15 cases that involved a single gene and 10 that included multiple genes. There is a very long supplemental table that includes all of the variants, but I just wanted to highlight the ones that were highlighted in the paper itself. So one was a 16p microdeletion. There was a 17p microdeletion that's also called Smith-Magnus syndrome, a TC4 deletion called Pitt-Hopkins syndrome, and a UCSC3 deletion. And then in terms of implications and what these findings mean for counseling, the identification of a molecular diagnosis allowed for our standard empirical recurrent risk of 1 to 2% to be revised. So if there was a de novo pathogenic variant, the risk was adjusted for those families to less than 1%. However, if it was a recessive variant, it was increased to 25%. And then if it was a dominant or X-linked variant, that increased to 50%, which was helpful for counseling those patients. So in terms of conclusions, genetic susceptibility in addition to number, duration, severity, and timing of insults affects neuronal, structural, and functional integrity. SNPs involved in inflammation and coagulation have been implicated as risk factors for cerebral palsy. And although cerebral palsy may result from multiple gene environmental interactions, the role of the individual components themselves remains unclear. And precise diagnosis is important for establishing causal pathways and enabling parents to plan for the care of a child and understand future risk of recurrence in future pregnancies. These are my references, and that's it. Does anybody have any questions?
Well done, Jen. I loved this particular topic. I was excited to see that you were presenting it because I think it's a topic that's not talked about enough. We often see a lot of the times, and I know that the genetic counseling students can agree, when we're taking a family history, CP comes up quite a bit. And of course, there can be due to several non-genetic reasons, but a lot of the time we might find that there might be more than one family member that has CP. That especially if they're siblings, it starts to raise suspicion. So we really, it's just a new and emerging area. And we actually did see a patient, I want to say like about a year ago, who came to us with CP, who came to us for preconception counseling. And we discussed the possibility of pursuing a panel to see if there was a potential underlying genetic etiology for her diagnosis. Unfortunately, the testing was put on the back burner because it was at the start of the pandemic. But through our research, we found that there was a panel at GeneDx that actually tested for over 1,000 genes, which on some level I have to laugh because at that point I would consider possibly just pursuing exome at that point. But yes, so I think it just really illustrates the point that the genetics of CP, if there is a genetic etiology, is quite complex, and I learned a lot, so I just wanted to thank you for that.
Stephanie, on that was going to be my question actually. Wonderful presentation. That my question was going to be, what panels, to send. Actually, a quick 1-minute comment. This is one of the things that has the potential to revolutionize really OB care, because there are CP cases and, it's been a black box so far. And I can tell you on the academic side, medical side, patient side, but also medical legal side, this has huge implications. A lot of obstetricians have been blamed in the past for something that, may have been very well genetics, maybe not all of it, but certainly a significant amount by all the studies that Jenani reviewed. So the question comes, is there a panel that we like the most? Is exome sequencing better? And I know there's no right answer, but what will be your opinion? I guess Jenani and then Stephanie.
In terms of panel, I think Stephanie, I'm not sure. And then I know cost is usually like the prohibitive factor with a lot of these things. But in terms of moving forward, it might become more something that we can do more frequently moving forward. I don't know if Stephanie knows about the panel. I'm not sure.
Truthfully, I don't know if many labs have this as a potential offering, except for GeneDx, and it does seem to be an area of interest as far as research for them. I may be mistaken. There was a talk at NSGC last year where they were discussing the potential genetic etiology of CP, and I'm pretty sure that a lot of the data was coming out from data found at GeneDx. So it could be like a passion project of that particular lab. But so I would say that at that rate, I would probably consider doing exome sequencing, just because I think it could be a little bit more comprehensive, just in case there was a particular gene, like a candidate gene that wasn't included on the panel that potentially could fit the particular phenotype, unique phenotype of the individual. That in that case, exome might be a better option. And what we're finding is that the cost of exome is starting to become a little bit more affordable. I say that a little bit more affordable because it still can be several thousands of dollars, but we're starting to find now that the cost is starting to be around the same as the cost of the panel. So in that case, I would pursue an exome for our patient if she were to pursue that test.
I don't know if some of our neonatal colleagues are on and want to comment. I know we usually also do neonatal updates. Does anybody want to comment from neonatology? They may not be on.
I don't see any of our neonatologists on that list, Vince.
Yeah, me neither. Yeah. Thanks, Jason. Any other comments from anyone?